Assalamualaikum and a very good morning everyone. Today our group would like to present a journal entitled Global Analysis of Bovine Milk Protein Variant Using Multi-Breed DNA Sequence Data. This study focuses on the genetic variation of major bovine milk proteins across multiple cattle breeds worldwide using DNA sequencing technology. So before we move into the findings of the paper, let us first understand the background of the bovine milk proteins and why this topic is important. In this slide, I explain the background of bovine milk protein and DNA sequencing. First, milk protein are very important because they provide essential nutrients such as amino acid, calcium, and phosphorus for calves. These proteins are also widely used in dairy products like cheese and yogurt. Next, bovine milk protein are mainly divided into two groups, which are casein and whey proteins. About 80% are caseins, while the remaining 20% are whey protein. These proteins are encoded by specific gene in cattle. However, these genes are polymorphic, meaning they have many genetic variants. In fact, around 60 variants has been reported, and these changes can affect the amino acid sequence of the proteins. Traditionally, scientists use methods such as electrophoresis, chromatography, and mass spectrometry to identify this variant, but these methods have limitations because they may fail to detect all variants and are mainly used for dairy breeds only. With the advance of DNA sequence analysis and whole genome sequencing, or WGS, scientists can now identify both known and novel milk protein variants more accurately across many cattle breeds worldwide. Overall, global genomic resources like the Thousand Bull Genomes Project and OptiBov project help provide a more comprehensive understanding of bovine milk protein diversity. However, despite the increasing number of studies on milk protein variant, previous research mainly focused on a limited number of commercial dairy breeds, particularly Holstein cattle. As a result, milk protein diversity in beef cattle and indigenous breeds remain less explored and not fully understood at the global level. In addition, traditional protein detection methods such as electrophoresis and chromatography may fail to identify certain variants accurately, especially rare and newly occurring variants. Therefore, with the advancement of whole genome sequencing technology, researchers now have the opportunity to investigate milk protein variation more comprehensively and accurately using DNA sequence data. Hence, the rationale of this study is to overcome the limitation of previous studies and provide a global analysis of bovine milk protein variant across multiple cattle breeds worldwide. This is important because milk protein variant can influence milk nutritional value, dairy processing properties, cheese making efficiency, and future selective breeding strategy in the dairy industry.
So the main objective is to investigate variation in milk proteins, which in a large number of cattle breeds based on whole genome sequencing data. Because why? Small DNA changes like missense mutation or splice site mutation can directly change the amino acid sequence of the milk protein, and this mutation can change the total amount of protein in milk or alter how proteins are modified after being made. And it can also directly impact the nutritional value of the milk. So it may affect the quality of the dairy products. Hence, for them, it is a great deal of interest in the identification of milk protein variants in cattle breeds.
To understand how the researchers uncovered these new milk variants, we have to look at their four-step bioinformatics pipeline. This process moved from raw biological data to digital sequence analysis, then to functional filtering, and finally to protein translation. Let's break down each stage. First, the team built a massive genomic library. They integrate data from the OptiBov project and the 1000 Bull Genome project, totaling 3,825 animals from 113 global breeds. For the new samples, DNA was extracted using GenTran kits and sequenced on the Illumina Nova Sequence 6000. This scale is critical because it ensures the study captures the genetic diversity of rare, native breeds, not just commercial ones. Second, they processed this raw code. The sequence were aligned to the ARS UCD 1.2 reference genome. They used Freebase for variant calling and Beagle 5.4 to infer haplotypes, which are groups of genes inherited together. To ensure accuracy, they applied a strict Fred scale quality filter of 20, meaning they only accepted variants with a 99% accuracy rate. The third step was filter. They used the Assemble Variant Effect Predictor (VEP) to analyze the 6 major milk genes. As shown in Table 2, they categorized millions of DNA changes to find missense variants, the specific typos that actually change the amino acid sequence. They cross-reference this against the DBSNP and EVA database. If a variant wasn't there, it was officially flagged as novel. Finally, the DNA was translated into protein sequence using the R package haplostats. They grouped these variations into final MIC protein bivariants. By comparing these two official dairy science nomenclature, they identified which were known and which were entirely new to the science. This rigorous four-step approach is what led to the discovery of the 86 novel variants we see in this study.
So the study analyzed 6 major milk protein genes and their surrounding regions, and they identified a total of 3,404 genetic variants, while half of its novel proteins, which is 1,660 novel proteins. When comparing milk genes to the bovine genome average, the research found that the missense variants, which change the protein structure, evolve at the rate of 2.8%, and it is nearly twice as fast as the genome average of 1.6%. And there's a massive 5 times increase in untranslated regions variation was also observed, and these regions are critical for controlling protein stability, while the majority of these findings are intronic, and the overall polymorphism rate of 3.56% prove that the milk genes are among the most rapidly evolved parts of the bovine genomes. By organizing these 59 mutations into adult types, we expanded the known library from 35 established variants to 121 unique proteins. Among them, 35 of protein variants are previously known and the famous site like the beta-casein A1 or A2, and also 86 newly discovered protein variants and categorized into common, novel, geo-specific, and extremely rare. And there are the summary of the known proteins which are detected and not detected. The known variants which are detected in the study shown in green color, known variants are not detected in this study are shown in white color, and novel variants described for the first time in this study are shown in gray color. Why were these 86 variants previously undetected? The reason is the traditional protein suppression has a significant resolution gap. Legacy tests like electrophoresis often fail to distinguish between the proteins with similar charge. For example, the beta-casein 1 was historically misidentified as A2. By using the whole genome sequence, we can read the DNA directly, allowing us to accurately discriminate between the look-alike variants and map the true diversity of genomes. As the conclusion, these 121 protein variants have direct real-world impacts, while the A2 family offers health benefits and beta-casein B is the gold standard for cheese yield and has better forming ability, but novel variants introduce new risks, especially the P149 phenylalanine mutation found in some novel variants destroy a phosphorylation site, and this could make the milk unstable during high heat industrial processing. And there are also other risk protein variants such as A1 variants, beta-casein A1 and 2. By understanding the library, this allows us to select for better health, better cheese, and better industrial reliability.
To wrap things up, we think that this was a massive global study because they didn't just look at local cows. They analyzed DNA from over 3,824 cattle across 113 different breeds all over the world. And the real game changer here was using whole genome sequencing, which allowed them to see details that older testing methods simply couldn't catch. And because they use advanced tech, they made some significant new discoveries. They found a total of 121 unique versions of milk proteins. And what's really cool about this is that 86 of these were brand new variants that had never been reported before. And it turns out that milk protein genes actually mutate nearly twice as fast as the rest of the cow's DNA. So this explains why there is such a huge variety in milk quality depending on where the cow is from. So why do you think we should care? First, because there are major health and nutrition benefits. Their findings help identify cows that produce A2 milk family, which is linked to better digestions and fewer stomach issues for people. Second, we think that it's a big win for the dairy industry because they found specific proteins like kappa casein, which are the gold standard for making cheese because they help the milk clot better and can increase the yield. So basically, this data gives us a map to produce better milk for everyone. To finish off, this study provides a foundational genomic resource because why? Farmers can now use selective breeding to intentionally raise cows that produce healthier milk or better cheese-making properties. And it really sets the stage for a new era of innovative dairy products.